Well, welcome everybody back to our Cerebral Blood Flow virtual seminar series. We've got our third session for this series for today, focusing on cerebral blood flow in hypoxia. Just a few notes again in terms of our rules for the road. So please keep your microphone on mute and your video off throughout the whole session. There'll be some time at the end of each presentation for questions. If you do have a question, please either use the chat feature and type your question, or you can raise your hand, which is under the participants link. The chairs will then ask you to ask your question, or they'll ask it for you if you've typed it into the chat feature. Just a reminder again that the session will be recorded and posted on the Cerebral Autoregulation Research Network website. And we have our first two sessions posted on there already. So please have a look at those if you haven't been able to make it so far. Just a brief note on the Cerebral Autoregulation Research Network. Please let me know if you'd like to join this organization. I'm the secretary, so just send me your CV via email and I'll add you to our list. So I'm very, very pleased to now pass off to the chairs for the session this morning. So Bara El-Khazraji will be one of the co-chairs together with Kurt Smith and Ryan Hoiland. So over to you, Bara.
Perfect, thank you. So thank you everyone for joining our seminar today titled The Regulation of Cerebral Blood Flow in Hypoxia from Mountainside to Bedside and Everything in Between. My name is Bara El-Khazraji. I'm an assistant professor at McMaster University. And I'm co-chairing this session with Kurt Smith, an assistant professor at Lakehead University, who will soon be joining University of Illinois at Chicago, and Ryan Hoiland, a postdoctoral fellow at Vancouver General Hospital and University of British Columbia, Okanagan. The theme for our session is brain blood flow and hypoxia, and we have a great lineup of speakers from 4 different countries, starting with our keynote speaker, Igor Fernandes, an assistant professor at Fluminense Federal University and University of Brazil. After that, Ryan Hoiland will be giving an extended abstract talk, as he will not only talk about his own research, but also provide further context to Igor's exciting findings. And finally, we have our 2 abstract speakers, Jie Liu, a research scientist at the Rutgers University Medical School, and Alexander Friend, a PhD candidate from Bangor University. If you have any questions, just a reminder to put that into the chat, and we'll do our best to answer as we go at the end of the session. And we hope that you enjoy our session. And without further ado, our keynote speaker, Igor, will now take control. Thanks.
Thank you, Bara. Thank you for the nice introduction. So just let me share my screen. Is everything okay? Are you guys seeing my screen?
Yeah.
Good, awesome. All right, so hello everyone. I hope you guys are all doing well. Maybe a bit bored by quarantine, but hopefully healthy and safe at home in the company of your beloved ones. I initially would like to thank Professor Dr. Brassard and Dr. Rickards for this wonderful idea of putting us together. And of course, to extend my gratitude to Dr. Hoiland, Dr. Smith, and Dr. Al-Khazraji for this wonderful opportunity to share with a so distinct audience part of the work I have been doing in the last couple of years. So my interest in the cerebrovascular physiology is pretty simple. I am just amazed how a special organ like our brain, who developed itself to have a size bigger than what's expected for human dimensions, 86 billions of neurons, what actually explains our notable cognitive abilities and separate us from other species, which also comes with a high energetic cost because they are highly dependent on the oxidative metabolism. But I'm really surprised how this organ evolved to be capable of storing minimal quantities of oxygen and nutrients. So it exclusively relies on the capacity, on the fine regulation of blood flow for its perfect functioning. So in other words, it's a very powerful organ, but very highly vulnerable to failure. And this vulnerability becomes evident when we think about in conditions where the flow is interrupted to the brain. So this, we all know the consequences. So in few seconds, we have loss of consciousness. And if it does take longer, probably will have strong clinical consequences and maybe neuronal death. So the question is, why did our brain not evolve to be able to survive anoxic conditions, such as the turtle's brain? So we don't have this amazing system that these wonderful animals do have, which allows a downregulation of the metabolic activity of the brain, allowing those animals to survive those conditions. But we don't need to complain for not developing like a turtle. We actually, even not being able to survive anoxic conditions, we are capable of adapting and performing very well in conditions where I would say there is a low availability of oxygen. And a great example of this capacity we have comes from this man, Mr. Reinhold Andreas Messner, which was considered the best mountain and best explorer in the world, because he was the first man who made the ascent of Everest without supplementary oxygen. And if you think about this accomplishment, it's actually amazing, because as reported by Professor John West in 1983, those incredible low values of barometric pressure at the summit of the mountain actually resulted, as reported later by the British group, in extremely low values of PaO2 and also arterial oxygen saturation. It means that somehow we evolved being able to adapt and perform very well in acute and chronic conditions in which we have low amount of oxygen available. And one of the aspects that explain this capacity is the ability that the cerebral vasculature has to deal with variations in the arterial oxygen concentration and precisely regulate cerebral blood flow and attend all those metabolic needs of this hungry organ. So when we are acutely exposed to oxygen deprivation, what happens is the blood vessels in the brain dilate. So this reduction in the vascular tone increases flow to the brain, which is not a very simple response, because it does depend on the increase in ventilation and the resulting hypocapnia, and also the sympathetic excitation resulting from chemoreceptors activation. But when you look at this interesting graph, which everybody knows because everybody uses in their presentations, you see a very clear and linear increase in cerebral blood flow in response to the reduction in the arterial content of oxygen. And this response is so effective that even in pronounced reductions in the arterial content of oxygen, it maintains stable, not only the cerebral delivery of oxygen, but also the metabolic profile of the organ. So as here represented by the small changes in the cerebral metabolic rate of oxygen. This powerful and effective vascular response seems to occur at the level, throughout the cerebral vascular tree, as we have animal evidence indicating vasodilation at the level of the resistance arteries, like the PO artery, the resistance vessels, like the PO arteries, at the level of intracranial arteries, like the middle cerebral artery, and most recently, evidence of the dilation, Doppler ultrasound and MRI human evidence of the dilation at the level of the extracranial conduit vessels, such as the internal carotid artery and the vertebral artery. So this response is also very complex because it seems to be characterized by regional differences. As you can follow the cursor here, the increases in flow in response to the reduced PaO2 seem to be greater in the posterior cerebral circulation, here represented by the vertebral artery, when compared to the anterior cerebral circulation, here represented by internal carotid artery. This is not a consensus, but there is MRI-based human evidence replicating these findings, but we still don't know what is behind, or what mechanism is behind these regional differences in response to hypoxia. Interestingly, we also reported regional differences, not to hypoxia, but also to the increase of hyperoxia, to the increase in arterial content up to the normal levels, a change that has been related to vasoconstriction and, as you can see in the curve, a reduction in cerebral blood flow. In this study, where healthy subjects underwent to isocapnic hyperoxia for 10 minutes under saline infusion. Different what has been reported to happen in hypoxia, the reduction in flow here represented by the ICA perfusion was greater in the anterior circulation when compared to the posterior circulation, which seems to not change under conditions of high availability of oxygen. We ended up doing a study in which we were interested in determining what was the mechanism behind this hyperoxia-induced vasoconstriction and reduction in cerebral blood flow. Pretty much the same thing. Healthy subjects underwent isocapnic hypoxia, hyperoxia, sorry, but not only under saline infusion, but also vitamin C infusion, because we hypothesized that hyperoxia would increase ROS production, reactive oxygen species, and this would consequently reduce nitric oxide bioavailability, generating constriction. In addition to blood flow measurements, we also were able to take some arterial and internal jugular vein samples to quantify not only indexes of ROS production, but also nitric oxide bioavailability. As expected, here represented by total cerebral blood flow response, perfusion in the brain was reduced during hyperoxia, which was mainly driven by a reduction in the internal carotid artery blood flow. Response that was also followed by an increase in arterial 8 isoprostane, which is an indicative of ROS production. That was also followed by an increase in transfeasible exchanges of nitrite, indicating that this response may be mediated by a reduction in nitric oxide bioavailability. So when subjects underwent hyperoxia under ascorbic acid infusion, we did not see that response, the reduction in blood flow. ROS production did not increase, and we saw a non-significant difference in TCE nitrite, indicating that at least for the hyperoxia-induced vasoconstriction, what happens is the increase in ROS production and the interaction with nitric oxide reduces its bioavailability, generating constriction and reducing blood flow. So this is a very straight scenario, but completely different from the scenario that we see related to the mechanisms of hypoxia vasodilation. This is a pretty chaotic, as you can see in the figure, but not only because we have several candidates, such as the release of vasoactive substances by the oxygenated erythrocyte, and also all the well-known endothelial-derived compounds such as nitric oxide, prostaglandins, and adenosine, but also because we have very few controversial studies investigating only the individual role of these endothelium-derived compounds, not considering the fact that they may affect the smooth muscle cells in a kind of, I would say, redundancy that would generate relaxation and result in hypoxic vasodilation. The only study that dealt with this redundancy was published last year by Dr. Williams Shiraz's group. They seem to be pretty successful to reduce or blunt the increase in the MCA conductancy response to hypoxia when reactive oxygen species and cyclooxygenase products were blunted simultaneously. So this to summarize the first part of my talk, it was clear that the human brain is vulnerable to hypoxemic episodes, but we do count on the hypoxic vasodilation, a compensatory mechanism that maintain adequate perfusion and oxygen delivery for its functioning. This response seems to occur through the vascular tree and also present regional differences. And the mechanism is still not very clear, but might be a consequence of mechanistic redundancy, although we are kind of seeing a light in the end of the tunnel. So in the first part of my talk, I kind of told you the story about the good relationship between evolution and the cerebral vasculature. But now we are going to move to a stage where I would say it's related to consequent bad consequences to the brain vasculature. And one of those consequences are the appearance of chronic disease. There is a special one which is highly prevalent around the globe, hypertension. As you can see in this beautiful map provided by the World Health Organization, hypertension is highly prevalent around the world, and it reaches a kind of status of pandemic as approximately 30% of the global population is hypertensive. And this is, has a very special impact in the brain, especially if you consider that hypertension is responsible for around 50% of all stroke-induced deaths we have in the world. Not only because of this, but also because the outcomes related to this condition, such as the increase in sympathetic outflow, the increase in sympathetic chemoreceptor sensitivity, and the cerebrovascular dysfunction, here represented by a reduction in the reactivity of CO2, they all raise the hypothesis that somehow hypertension may affect the capacity that the cerebrovasculature has to deal with oxygen variations in control flow. And this is of, I would say, a clinical importance, because these population is under increased risk for events of sleep apnea. So we ended up conducting a study trying to answer these following research questions. Arterial hypertension impairs cerebrovascular and peripheral reactivity to hypoxia. Is the sympathetic overactivation responsible for the likely deterioration of vascular reactivity to hypoxia in this population? So in this study, normotensive and hypertensive subjects underwent normoxic and isocaponic hypoxic trials, and we were able to simultaneously quantify not only cerebral blood flow, but also peripheral blood flow, and at the same time quantify muscle sympathetic nerve activity throughout the study. As you can see in the figure, we were able to evoke similar levels of desaturation in both groups, and at the same time maintain PET CO2 levels pretty stable throughout all the trials. And when we look at the local CBF responses to hypoxia, it's very, very clear that normotensive group showed a greater increase in both ICA blood flow and VA blood flow, but it only reached the statistical significance when we compare changes in the posterior cerebral circulation. As you can see in the figure, both groups increased total cerebral blood flow in response to hypoxia. But again, the same scenario when we compare the groups, normotensive subjects presented a higher increase in TCBF, which guaranteed an increase in the cerebral delivery of oxygen in this group, while although the response, the hyperperfusion response was impaired in the hypertensive group, the cerebral delivery of oxygen was pretty stable. But we have pretty much the same scenario when we compare the changes between groups, showing that that response was kind of impaired in the hypertensive subjects. When we look at the peripheral blood flow responses, scenario is pretty much the same. There is an increase in femoral artery blood flow in the normotensive group in response to hypoxia, but there was no change in blood flow response to hypoxia in the hypertensive subjects. Same scenario when we compare the groups, and when we come to see the LDO responses, the leg oxygen delivery, pretty stable in the normotensive subject, but there was a reduction in response to hypoxia in the hypertensive subject, which was also reflected when we compare the changes between groups. So we were also able to replicate the findings from Dr. Aboud's lab in 1998, where he also showed that hypertensive subjects presented a higher increase in muscle sympathetic nerve activity in response to hypoxia when compared to the normotensive subjects that also present a small increase in that variable. And to get insights about possible mechanisms, we correlate the changes in TCBF and also femoral artery blood flow with the changes that we observe in muscle sympathetic nerve activity. As you can see on the right, those changes were correlated in the femoral artery blood flow, and it seems that somehow sympathetic overactivation may play a role in this impairment observed in the hypertensive subjects. So with this, we conclude that hypertension does impair hypoxia-induced vascular reactivity in both the brain and the leg circulations. And from the correlations, we think the hypoxia-induced sympathetic excitation contributes to the impairment of the peripheral vascular reactivity, although we cannot discard the contribution of this sympathetic overdrive in the brain response. So inevitably, after the study, we came with the following research question. What was the mechanism behind this impairment in the brain circulation reactivity to hypoxia in the hypertensive group? So aware of the likely role of nitric oxide in this hypoxic vasodilation, we also saw a defective nitric oxide pathway in a hypertensive postmortem model in which acetylcholine, instead of vasodilation, induced contraction in the vertebral arteries from treated hypertensive patients. So Laura, a very good friend of mine, came with the wonderful idea to see how the hypertensive cerebral vasculature would respond to L-arginine infusion, which is a precursor of nitric oxide, and it's a well-known vasodilator. So we ended up submitting normotensive and hypertensive subjects to either L-arginine or saline infusion for 30 minutes. Just a quick reminder, we were not able to quantify ICA blood flow in that time. So we did quantify the common cerebral blood flow. That's why we don't have calculations of total cerebral blood flow and also oxygen delivery. As you can see in the graph on the left, L-arginine infusion evoked a huge increase in blood flow in the common carotid artery, which was similar in both groups. As expected, blood flow in the carotid artery did not change with saline infusion, but as we hypothesized, it seems that the response in VA blood flow to L-arginine infusion was blunted in the hypertensive patient subjects, while there was a greater increase in that response observed in the normotensive group. As expected, again, saline does not change VA blood flow, and these findings made us come to the conclusion that probably a defective nitric oxide pathway is a candidate to explain the impaired hypoxia-induced vascular reactivity in the brainstem circulation of hypertensive men. So I was pretty happy with the story and with the happy end as well. But in one of my random readings, I found out that there was a specific isoform of potassium channels at the smooth muscle cells that may contribute to the hypoxic vasodilation, especially in animals. So this isoform was the isoform ATP sensitivity isoform, which in this interesting study published in 1994 showed that a cerebral vasodilation to hypoxia, as you can see in the white bars, was dramatically reduced under the presence of glibenclamide, which is an antagonist of those KATP channels. And more importantly, we found out that the KATP channels functioning was kind of impaired in hypertensive animals, as cerebral vasodilation was not so effective as you see it in the control animals during the infusion of apricoline, which is an agonist of the KATP channels. So we went to the human literature. There was no report about the possible role of the KATP channels. And we then were pretty excited to translate those findings to humans and answer the following research questions. So do KATP channels mediate increase and maintenance of cerebral oxygen delivery in humans under hypoxia? And also if the arterial hypertension would impair their role in that phenomenon. So due to personal issues and also COVID-19, we were not able to answer the second question, but we did answer the first question with the most recent paper we published in the Journal of Physiology, which was about the role of KATP channel and the isocapnic in the response to isocapnic hypoxia in humans. So in this study, we have a pretty simple model in which we submitted healthy subjects to normoxia and isocapnic hypoxia before and 3 hours after the glibenclamide ingestion. So in this study, we were also able to quantify blood flow, the local blood flow responses simultaneously, and also take some radial artery blood samples to quantify oxygen delivery. And as you can see, we were pretty successful to replicate those findings in animals. As expected, before the glibenclamide, the response was pretty clear. We had an increase in the anterior cerebral circulation blood flow, which was blunted 3 hours after glibenclamide ingestion. When you look at the differences, the percentage differences in that response, there was a very big attenuation in the hyperperfusion response in the ICA as a consequence of the KATP blockade. For our surprise, nothing really happened in the VA response. So as you can see here, the response was preserved in both conditions, and it was reflected when we compare the changes induced by hypoxia. So that reduction in ICA blood flow was also reflected in the TCBF response, which was preserved during hypoxia before glibenclamide ingestion, and was actually blunted at 3 hours after the ingestion of the drug. Again, when you compare the changes between conditions, it seems the KATP blockade was pretty effective with a huge attenuation of that response during hypoxia after glibenclamide. CDO2 somehow was pretty stable in both conditions, although we have a small non-significant decrease in the glibenclamide ingestion, but somehow when we compare the percentual changes, glibenclamide was able to change the profile of the response and indicate significant difference between conditions. Our data was also supported by a condition where we submitted those subjects to the same protocol, but now before and after placebo. And as you can see here, for ICA and for VA, the response was preserved before and after 3 hours placebo, which was also reflected for when we compare the changes between conditions. TCBFs and CDO2 had pretty much the same response, although we find out that after placebo, hypoxia induced a greater increase in TCBF, at least for the absolute changes, which was not reflected when we compare the percentual changes between conditions. And as expected, CDO2 did not suffer any significant changes when compared between conditions. So based on these findings, KATP channels do mediate the increase in local and total cerebral blood flow and seems to contribute to the maintenance of cerebral oxygen delivery in humans under hypoxia. Just to finish, it seems that the human brain is vulnerable to hypoxic episodes. I mentioned the compensatory mechanism that maintain adequate perfusion. It seems that somehow ATP-sensitive potassium channels in both animals and humans are involved in this response, and hypertension affects the compensatory response and seems to affect that via impairment nitric oxide pathway. In the future, I hope we'll be able to respond if there was an impairment in the function of the KATP channels as well. Just before the finish, some rapid acknowledgements for all my collaborators around the world. Special thanks to Dr. Secker for all the help in the studies. Without him, it wouldn't be possible. And also a special thank to Dr. Norberger for all the opportunities that and the support that he has given to my career. And to finish, I would like to thank this team, which was responsible for all the experiments. It was a pleasure to work within, and also all the financial agencies that supported my work. Thank you very much, and I'll be happy to take your questions.
Thank you, Igor. That was a great talk. Very impressive. We've got our first question. I'm not sure if you can see it, but I'll ask it. So this is from Binu Thomas. How do you explain the ascorbic acid base increase in cerebral blood flow on hypoxia?
Well, I'm not sure if I got the question. We didn't see an increase in cerebral blood flow during the vitamin C infusion. We were actually trying to block the production of ROS, reactive oxygen species, which are consequent of the hyperoxia of the increase and the arterial content of O2. Does that answer the question?
I think so. I think you're right with the data you showed. Another question from Wabi El Bouri. Thank you very much for the talk. What he's interested in is normotensive people seem to overcompensate with their cerebral blood flow response, increasing arterial oxygen to increase arterial oxygen delivery. Hypertensive people do not have as much compensatory responses, but they still have a healthy delivery of oxygen. So what is the cause? What causes hypertensive people to be more prone to disease such as stroke or dementia? Do you think it's a prolonged lack of overcompensation that may cause long-term structural responses, or it's something else?
Well, it's hard to answer, but I'll give you my perspective based on the data that we have. Those hypertensive subjects were pretty young. So that response might be a consequence of this profile. They did not have any kind of other, I would say, problem. They were only hypertensive. We had the care to see if they had any other problems, because we wanted to just, identify the role that hypertension plays in a likely impairment of vascular reactivity. So that's why I think that response was reserved. But if you probably take another group of hypertensive subjects, which has like the other aspects like age, obesity, probably you have an impaired response. So that's why I think we had this preserved CDO2 response during hypoxia.
Yeah, I was wondering myself a little bit about that, Igor. Considering some of Emma Hart's work with the selfish brain hypothesis, did you guys screen for any differences in vertebral artery diameter on either side?
That's a great question. We did not. We actually were expecting a reduction in blood flow, especially in the vertebral artery, especially based on the response on the selfish brain hypothesis. But one thing is important to say, that all these subjects, they were not under medication. So it seems that when they are not under medication, the increasing pressure guarantees the appropriate perfusion, and that's the basis of the I would say the selfish brain hypothesis, because pressure increases just to maintain the appropriate perfusion in the brain based on the Cushing response, right? So as those guys, they were not under medication, we did not see any differences in perfusion. But just to answer your question, we are not interested in possible differences, anatomical or functional anatomical differences between those groups.
Excellent. Probably gonna take one more question, Igor, and then we'll move along. There'll be some more questions in the comments if you feel like commenting after that. But
yeah.
The next question was from Alex, and he's asking how total cerebral blood flow is measured in your subjects. I wonder if you just missed that one slide.
Yeah, I'm sorry, I probably missed that point. We quantified simultaneously blood flow responses in the internal carotid artery and the vertebral artery. So when you sum and multiply by 2, because we have a pair of both arteries, you get a total cerebral blood flow response.
Excellent. Well, we'll hear more about that, I think, in Alex's talk, I think, too, later on here.
Okay. All right. Thank you very much for your attention.
Yeah, thanks, Igor. That was very impressive. Great start to the symposium. We're going to
Thank you very
switch
much,
over
Greg.
now to Dr. Ryan Hoiland. You ready, Ryan?
Yeah, 1 second. Awesome. Can you see that? Thumbs up, Kurt. Perfect. Awesome. Tough act to follow. Fantastic presentation, Dr. Fernandes. So the title of my talk today is called, Are We Stuck Scratching the Surface of Hypoxic Cerebral Vascular Control in Humans? The answer is no. So just to retouch on a couple of the basics of brain blood flow control and hypoxia, I have up here 2 axes, an x-axis with arterial oxygen content, or CaO2, and a y-axis with cerebral blood flow, or CBF. And we've known for quite a long time, especially considering a few studies done in the late 60s and early 70s, that hypoxia increases brain blood flow. And this has been replicated in a multitude of studies more recently, including one from my PhD supervisor, Professor Phil Ainsley, as well as the research by Dr. Fernandes. And the implications of this are that cerebral oxygen delivery is maintained during hypoxia. And so from this, a prevailing research question in this area is, we know that this response works, we know it's robust. What are the mechanisms that are actually causing this response and allowing this increase in cerebral blood flow to maintain oxygen delivery? Now, as we saw, Dr. Fernandes reported on some data most recently in his KTPA channel blockade study. And what I've put up here now in the star symbol is just his control data. And we see that he shows the same cerebral blood flow response that's been previously demonstrated. But following glibenclamide, as he demonstrated, this is reduced. And the consequence of this is that cerebral oxygen delivery is now no longer maintained. So reading these results, I thought to myself, why is it that we saw that Dr. Fernandes saw this reduction in the brain blood flow response and reduction in oxygen delivery with glibenclamide, when historically studies in humans that have attempted to replicate animal model results have failed to demonstrate effects? And so I'll touch on that a little bit here. And so part of this thought was that perhaps a lot of this derives from us scratching the surface, so to speak, where we're independently blocking either bloodborne or endothelial mechanisms of vascular control, such as nitric oxide mediated vasodilation. We have S-nitrosylethyl or RSNO release from red blood cells, nitrite, shear stress, and ATP binding of purinergic receptors that may all increase NO bioactivity. But thus far, we haven't seen reductions in brain blood flow responses to hypoxia, or they've been inconsistently demonstrated with nitric oxide blockade. Other vasodilators such as prostaglandin, adenosine, and ATP have also independently not shown an effect on hypoxic vasodilation in humans. We do now know from the study from the group from Wisconsin that combined cyclooxygenase inhibition and reactive oxygen species inhibition that causes a reduction. And now the study from Dr. Fernandes's lab. And so one of those reasons might be that KTPA channels specifically block the convergence of a multitude of pathways. And so in this figure, which is a bit chaotic, there's black lines representing select vasodilatory pathways, and then green lines highlighting the components of these pathways that converge on the KTPA channel. And we can see that by blocking KTPA channels, we would be blocking quite a bit of vascular signaling pathways. And so perhaps it's that because we've been focusing on these surface level mechanisms, we haven't been able to detect changes in vascular control because of all of these redundancies within the vascular smooth muscle. And this may be the step forward, looking at these redundancies as has been demonstrated by Will Schrage and Dr. Fernandes's work. And so this question of, are we stuck scratching the surface, is what I'll talk about today with some research we've done in our lab at UBC Okanagan. And as denoted in the title, the answer is no, and I hope to convince you of this as we move through this talk. And so the aims and hypotheses for the research that I'll present is 1, to determine the role of nitric oxide in regulating hypoxic cerebral vasodilation in humans, and 2, to determine the role of the red blood cell in regulating hypoxic cerebral vasodilation, because we know that the red blood cell has a unique role in conveying nitric oxide mediated bioactivity following its deoxygenation and circulatory transit. And our hypotheses are that pharmacological inhibition of nitric oxide synthase would reduce the cerebral blood flow response to hypoxia, and that red blood cell release of S-nitrosethiols, which are these NO species released from the red cell during deoxygenation, would be related to the magnitude of hypoxic cerebral vasodilation. So to address these hypotheses and aims, we conducted 2 studies. The first of which we looked at pharmacological inhibition of nitric oxide synthase using the competitive and non-selective inhibitor Ng-monomethyl L-arginine, or LN-MMA for short. We conducted this study in 11 healthy males with an average age of 25 and a BMI of 24. Now for this study, we conducted what I'll refer to as an isocapnic hypoxemia test. So in 2 sequential stages, we reduced arterial oxygen content by reducing end-tidal O2 and then saturation to 83 and then saturation to 74, meanwhile maintaining isocapnic conditions to isolate the influence of hypoxia on cerebral vascular control. We then conducted our LN-MMA infusion protocol, where following a bolus infusion, a maintenance dose is started and a minimum 30-minute wash-in period was allowed prior to reconducting the same isocapnic hypoxemia test. And you can see that we were relatively successful in matching the severity of hypoxia between these 2 trials. It's also important to note that while I have them demonstrate sequentially here, the order was randomized and counterbalanced, and we provided an adequate amount of time for drug washout between trials. Now for study 2, I want to provide a little bit more background. And so thinking about hypoxia, I want us to think about it now in 2 different ways. One, hypoxemia, referring to reductions in the partial pressure of oxygen and hemoglobin saturation, which would theoretically engage allosteric hemoglobin-dependent mechanisms of vasodilatory control. And then anemia, or as I'll refer to hereafter as hemodilution, reduce red blood cell concentration and lower hemoglobin levels that then reduce arterial oxygen content. To display these 2 types of hypoxia graphically, I have a figure here with arterial oxygen content on the y-axis and the partial pressure of arterial oxygen on the x-axis. And so this isn't specifically the oxy-hemoglobin dissociation curve, but it looks the same because of the linear relationship between content and saturation. And I presented this relationship for individuals with a hemoglobin of 14 and arbitrarily selected an individual with a hemoglobin of 10. The takeaway from this is that if we have an individual with a normal hemoglobin level and we reduce arterial oxygen saturation to around 70%, this is going to lead to large reductions in arterial oxygen content. However, if we maintain saturation in individual and we reduce their hemoglobin concentration and make them anemic, this also can lead to reductions in arterial oxygen content. And we can use 2 different stimuli to interrogate the cerebrovasculature differently, but with an overall maintained hypoxic stimulus. And the reason we did this was to attempt to specifically interrogate allosteric hemoglobin changes by desaturating hemoglobin or allowing for maintained saturation in the arterial circulation with anemia. So for this study, we tested 10 healthy males aged 29 years with a BMI of 23. And our first experimental protocol was a hypoxemia trial where we conducted 3 stages of isocapnic hypoxemia, maintaining CO2 as previously mentioned. Following this, we conducted our hemodilution protocol, where in 2 sequential steps, we removed and replaced 10% of whole blood volume with 5% human serum albumin. So this led to 10 and 20% reductions in hemoglobin concentration that we see here. And then finally, following this hemodilution protocol, we repeated an isocapnic hypoxemia protocol again, utilizing the same reductions in arterial oxygen saturation. But what we'll note here is, of course, content as denoted by the height of the bars is lower at every stage comparative to the pre-trial because these individuals' hemoglobin concentration has now been reduced. The experimental measures we used for these two studies were radial artery catheterization for blood gas measurement and collection and storage of blood for later nitric oxide analysis, and then jugular venous catheterization. And we can see here that the jugular venous catheter is advanced to the level of the jugular bulb, which is approximately at the level of the top of your ear. And this allows for us to selectively sample cerebral venous outflow. We use this for blood gas analysis as well as collection and storage of blood for later nitric oxide analysis. To measure cerebral blood flow, we used duplex ultrasound of the internal carotid artery. As we can see in this top ultrasound image, we have the longitudinal view of the artery here and the corresponding waveform, as well as in the vertebral artery, where again, we have the cross-section and the velocity waveform. And then we estimate global cerebral blood flow as twice the sum of our internal carotid and vertebral artery measurements. With our cerebral blood flow data, we calculate what we refer to as a reactivity profile, which is the unit change in cerebral blood flow per unit change in arterial oxygen content. And so just to reiterate, I have graphically demonstrated the relationship between cerebral blood flow and arterial oxygen content, whereas content decreases, cerebral blood flow goes up. And we can use linear regression or other linear analysis techniques to assess the slope of this relationship, which is considered the responsiveness of the cerebral vessels to hypoxia. Looking at the results from these two studies, I have global cerebral blood flow reactivity on the y-axis here. So a higher number on the y-axis is a steeper slope. And we can see that compared to our volume matched saline placebo trial, LNMA led to a 23% reduction in the blood flow response to hypoxia. Now the implications of this for cerebral oxygen delivery are denoted in the second panel, where we can see that as arterial oxygen content decreased in the saline trial, there was a small but linear increase in cerebral oxygen delivery. However, this was effectively eliminated with LNMA, where there's an 85% reduction in the oxygen delivery response to hypoxia. Moving on to our hemodilution study, I again have cerebral oxygen global cerebral blood flow reactivity on the y-axis here, pardon me, and we have our hypoxemia trial in red, the reactivity to the staged anemia or hemodilution in the blue, and we can see that this reactivity was reduced by 45%. And then reactivity to hypoxemia following the hemodilution protocol, we can see that reactivity here was actually effectively doubled or increased by 100%. Excuse me. Looking at the implications for cerebral oxygen delivery, we can see that in the hypoxemia trial, there was an increase in cerebral oxygen delivery as content was reduced. However, in the hemodilution trial, this oxygen delivery response was reversed. And at the end of the hemodilution protocol, delivery was actually reduced by 5%. And then in the hemodilution and hypoxemia protocol, there was again a greater delivery response than in the original hypoxemia protocol. Moving on to some nitric oxide analysis results. We used ozone-based chemiluminescence to assess different nitric oxide species in the arterial and jugular venous blood. I'll just present today, for simplicity, the S-nitrosylethyl data within the plasma. And what I have represented up here is the arterial to venous difference. And so a negative number here indicates release. There's a greater amount of nitrosylethyls in the venous blood than in the arterial blood. And a release here would be theorized to indicate a release of nitrosylethyls from red blood cells, which would then subsequently induce NO-mediated vasodilation. Now, if we look at our hypoxemia protocol, we can see that where there was no gradient in normoxia, we get a statistical negative gradient here, or RSNO, nitrosylethyl release. However, in the hemodilution protocol, this release was not observed. And if anything, the gradient went in the other direction. But again, during the combined hemodilution and hypoxemia protocol, there was a negative S-nitrosylethiol gradient indicating potentially release from the red blood cell. Now, to relate this back to the global cerebral blood flow values during each of these conditions, we have the lowest global cerebral blood flow during normoxia, as expected, the increase in global cerebral blood flow during hypoxemia, but again, during hemodilution, that increase in blood flow is lower. And it's important to note that these blood flow measurements and S-nitrosylethiol measurements for these two specific conditions are at a matched arterial oxygen content or hypoxic stimulus. And then blood flow is the highest during the combined protocol. Now, to assess this further, we conducted a within subjects repeated measures correlation analysis. And for our group, there was a negative and significant relationship between the arterial venous S-nitrosylethiol gradient and cerebral blood flow, indicating that across all these stages, a greater release of S-nitrosylethiol was related to a higher cerebral blood flow. Now, to bring this back into context with the results we recently saw from Dr. Fernandes, I've replotted just the weighted mean from a multitude of studies assessing cerebral blood flow and hypoxia. And then I've overlaid Dr. Fernandes' results with the post-Glevencomide data with being the open stars. Now, if we add the data from our recent studies here, you can see that pre-hemodilution, there's a normal response and an increase in oxygen delivery. During the hemodilution protocol, there was a blunted cerebral blood flow response and a reduction in cerebral oxygen delivery. In our saline trial from our LNMMA experiment, again, we see a normal blood flow and oxygen delivery response. The following LNMMA, we again see a blunting of this response and no increase in cerebral oxygen delivery. And so we can see that prior to and following these blockades, there's definitely a distinct cerebral blood flow response with distinct implications for cerebral oxygen delivery. And with that, I'll conclude my talk. I have many individuals to thank. These were two large projects. First, I want to thank Dr. Caroline Ricards and Professor Prashis Prasad for organizing this seminar, and my PhD and postdoc supervisor when we conducted these studies, Professor Phil Ainsley, and then this was a large team effort, so thank you.
Well done, Ryan. Great. We'll open up the floor to questions. I got a quick one. So when do we get to see the first double or triple block study?
Oh, once I get a professorship and a lot of money to do it. I don't know.
Right.
It's definitely the next step, I think.
Did you guys notice any blood pressure changes with the hemodilution?
No, blood pressure wasn't substantially altered. I can't quite remember, but the differences in the blood flow responses we see are mirrored by differences in cerebrovascular conductance changes, accounting for blood pressure. And also, if you include cerebral perfusion pressure, mean arterial pressure minus internal jugular venous pressure as a statistical covariate, there's no difference. There's the differences I showed today remain. So that would mean that these different responses aren't being driven by blood pressure.
We got another question here from Sandra Billinger. So for the global blood flow calculation, how do you account for blood flow delivery to the cerebellum, particularly when looking at or considering the vertebral artery flow?
Yeah, good question. I would just say that like currently this is, yeah, I'm not too sure. We can't really specifically account for anything locally with these two measurements because they are, despite being anterior and posterior, relatively global measurements themselves. And so, you know, reproducing a study like this in an MRI would be interesting to look at whether or not there's more specific regional variations, and that's also a future goal. Now that we've demonstrated these effects, is delving deeper into what they mean for local vascular control. I realize I didn't answer your question, but I don't have an answer, so apologies.
I would probably not 100% accurate to say, but I mean, perhaps given that it's supine and that they're not engaging in any form of activity, maybe the change in cerebellar flow isn't going to be affected. But as soon as you say that, I doubt it. We got a question from Andrew Robertson. Fascinating study. How strong was the within subject correlation between changes in cerebral blood flow and changes in arsenal?
So what we did is we conducted a repeated measures correlation. So it allows us to look at effectively the within subjects correlation, but across an entire group. So it fits the same slope to all the subjects. And it was, we had a R squared of negative 0.57.
We're going to give one more question here. We've got AJ Rosenberg. Do you expect these results to be similar in females?
Yeah, good question. So that's one of the other things that we want to do to follow up. I'm not sure about either of them, but we have some data to suggest that there may be a difference in the relationship between hemoglobin and hypoxic reactivity in males and females. In the data I showed you, there was an increase in reactivity once the male subjects were hemodiluted. We have data in, I believe, about 75 males looking at just across individual differences in normal hemoglobin concentration, so no intervention. And those with a lower hemoglobin concentration have a high reactivity, similar to what we showed with our within subjects design. But in females, of which I believe we have 60 data on 60 individuals, there is no relationship, or we don't demonstrate a relationship between their hemoglobin levels and vascular reactivity to hypoxia. So it's definitely a next step for us.
Awesome. And there's a couple more really good questions in the comments if you feel like answering those. But thanks for that awesome talk. And as Dr. Schrag says, a really amazing and rigorous study design. Great to see it.
Awesome, thank you. I'll get to the comments now.
Jie, are you ready?
Yes, I'm gonna start to show now. Everything looks good?
Looks good.
Hello? Okay.
Yeah.
So let's begin. So hello everybody, and very nice to present our studies on Tibetan's CBF here. So which is mostly based on our two published papers. So I started Tibetan CBF because 10 years ago, I had joined a medical aid team to work at Tibet for a whole year. And before that, I was a radiologist on ultrasound. But this experience at high altitude really got me love the study on hypoxia and the integratory physiology. So later, I finished my postdoc training at Dallas, and now I'm a research scientist. So although I'm kind of suffered from hypoxia there, but I really enjoy the life there. Here are my personal photos I took. I love the beautiful landscapes and very nice people there. However, some people can really get mountain sickness. And my 1 year close working with the medical team members much enhanced my knowledge in altitude medicine. So the reason for study on Tibetan is that they had lived with hypoxia for thousands of years. And they have the closest genetic similarity with Han Chinese. So thus we naturally have a group to study the genetic adaptations to hypoxia. Tibetans are demonstrated to be better adapted to hypoxia. So 10 years ago, scientists already screened out some related genes. And very recently, more works were published regarding their links to the functions of phenotypes. So then why study CBF? Firstly, because my clinical experience made me doubt the reported higher CBF in Tibetans. Secondly, I felt more confident to repeat some studies with my strengths. So thus we performed the first study, which was a cross-sectional design with over 300 subjects. All were young and healthy. So here shows how ultrasound measures CBF, which is now very popular now. We added a CBF of bilateral internal carotid and vertebral arteries to get the total CBF. Here shows how the ultrasound images looks like. So before the data interpretation, so please note the difference between the terms of acclimation and adaptation. So my simple thinking is that to check if the changes are temporary or permanent, even without exposure. So that's a simple way, I think, to understand. So first, let's just see the results from Han Chinese groups. Because in this group, so we also included a group of 2 day. You can see here a 2-day group. This group represents a group before the acclimation. So for easier understanding, please assume this data like a longitude data. So from the lives, you can see this is the sea level, Han Chinese groups at sea level. That assume the people go sea level to the high altitude. This is for 2 days, 1 year, and 5 years. So we can from this graph we can estimate how people will change their total cerebral blood flow during the long-term acclimation. So we can see here the results of 2-day group is consistent with previous longitudinal studies for accurate response. That's a typical response before the acclimation. So, but a low longitudinal study can be performed for longer than 1 year follow-up. So my data shows that after 1 year, there's no difference between 1 and 5 years. Even after acclimation, the CBF come back to the sea level, but there's no difference between 1 to 5 years. So, and we didn't see sex difference in this data. However, sex appears to be in the oxygen delivery, which is reason because the female always have a lower hemoglobin concentration. But you can see more interesting is oxygen delivery. So all oxygen delivery is displayed in as yellow color. So you can see the oxygen delivery actually didn't change. Even before the acclimation, it's still not too much change. So this kind of cannot be changed by acclimation. So this part, this metric. So next we come to the ratio comparisons. So note in this data, we combined the 1 and 5 year groups into one Han Chinese group to represent the group after acclimation. So please just see the red cross marks to check the racial difference. So the red cross only refers racial difference. So we can clearly see lower CBF in Tibetans, no matter female or female. So, but this difference not shown in the sea level, low altitude. There's still no sex difference here. More interesting, Tibetan's oxygen delivery showed a lower at both. You can see here, Tibetans, note the red cross. Tibetan's oxygen delivery showed both lower at both low altitude and high altitude. So this typically, so all these findings indicate Tibetans have a unique CBF regulation pattern. So in order to confirm these findings, we performed a simple experiment to study Tibetan's CBF response to oxygen inhalation. all subjects were young and healthy, but only males. So the protocol is we start resting baseline data collection for 3 minutes. And then we begin the oxygen inhalation. So the first 4 minutes, we don't do data collection. We only do the data collection after 4 minutes. And so after 4 minutes, we collect the data collection for 3 minutes for the post-oxygen data. So this is actually to reply the static response of the oxygen inhalation. So here shows how the lab looked like. And the left shows the typical TCCD image, which is kind of different from the traditional TCD. Here shows the values of oxygen saturation and CBF velocity. So you can see only the baseline of the 3-day group before acclimation, that's the only difference to other groups. But you can feel that the slope looks like different from different groups. So, we are more interested in the slope difference, which reflect the response. So note, although we can't get the absolute value of oxygen delivery, but the change of it actually can be estimated if we assume our CBF difference can be represent the total CBF difference. So here, please also note pay attention to the red pound sign, because only red pound sign reflects the racial difference to the Tibetan group. So we can clearly see the racial difference in oxygen delivery compared to the both 3-day and 5-year groups. However, the acclimation seems to have some impact on the COD, on the oxygen delivery and CBF. You can see the difference between before exclamation and after exclamation. There's some difference. So one concern is that the subjects had a individual difference in oxygen saturation change. So I feel necessary to normalize these responses to the oxygen saturation. So the red panel shows the value normalized by oxygen saturation. So there's more interesting finding. So you can see both CBF and oxygen delivery response are very consistent now. So both of them are different from Tibetans. So this reflects a kind of change different in the adaptations. So the take-home message is Tibetans blunt their CBF response to oxygen. So by then, many thanks to the funding agencies and all the people who helped me in my scientific research. With that, I'm happy to take any questions. Yeah.
Thanks, Jie. It's a great talk. There was a question during your talk by Alex Razumowski, and I think it was for everyone speaking, that we need to remember or make the distinction between how brain blood flow is measured in either milliliters per minute per 100 grams versus centimeters a second.
Yeah. Okay, thanks.
Jie, I've got one quick question.
Yes.
Considering the racial differences seem to be in total oxygen delivery and not blood flow, if we move back to the men and women comparisons from the previous study, what do you think the response would be if we kind of did the opposite of Ryan, but tried to increase hemoglobin or hematocrit in the women? Would we see a differential blood flow response, given that we may elevate delivery by just increasing the content?
Sorry. Yeah. So you said increase. Oh, you said the oxygen delivery. But yeah. So your question is a good question. So your thinking is about if the concentration hemoglobin will have impact, right? Have the CB.
Yeah, I'm wondering if we could control for the sex difference in delivery by changing hemoglobin concentrations. And that would maybe impact flow. So that might tease out some of that mechanism that you saw in the men versus the women for flow. And then I'm wondering then, would we see a sex difference and race difference in your secondary study?
Yeah. So yeah, that's a good question because we need but this all these measures is connected because when you make assume that female and male have similar hemoglobin, but that data will be not represented because they cannot, because women always have lower hemoglobin, so you can, so that's just my consideration.
Yeah. There's another question here. This is from Giri. I appreciate the distinction between acclimation and adaptation. However, they're wondering if you could provide a little more context to how your results impact acclimatization or adaptation.
So into the somebody if you help me more specific, because seems to me it's not so these questions what in the because first I admit the limitation because this is only a cross-sectional, so we can actually only follow. Ideally, we need to follow up more closely to see how this changes. But this is not that study design, so it's hard to explain for that.
One more question. Did you consider or measure at all carbon dioxide?
Yeah. That's a big limitation because we, because at that time is we just leverage the current available resources, so we didn't have that that time. So, but it's a good question. But actually, some data I checked. Some data shows actually Tibetans have a yes. This is a good question. Yeah, our limitations.
Okay. Well, thank you very much. Impressive research and great presentation. We're going to move on now to our final abstract. We're going to get some insight into the measurement of brain blood flow. Alex, are you ready?
Yeah, just give us a second. And hopefully, can you see my screen?
All good, mate.
Brilliant, fantastic. Well, hello, my name is Alex Friend. I'm a second-year PhD student at Bangor University. I'd like to thank the organisers and the chairs for inviting me to talk today about my first project, the extracranial regional cerebral blood flow regulation in hypoxia, a left and right side assessment by Doppler ultrasound. So we know that hypoxia stimulates this large increase in blood flow to the brain, and on a tissue level, when investigated by MRI, there's this variation in blood flow response to hypoxia with increases and decreases across the tissue. Now, whether this is mimicked at the level at the neck with the large arteries, the internal carotid arteries that feed 75% of blood flow to the anterior regions of the brain, and the vertebral arteries that feed 25% of blood to the posterior regions of the brain, remains to be seen. Now, it's usually determined through sensitivity or reactivity and the change in blood flow response to hypoxia. But using absolute values of increases in blood flow to hypoxia in these vessels is misleading because they're highly dependent on this 75-25 difference in baseline flow. They're bigger. Internal carotids are larger, so it's going to be highly dependent on baseline flow. So it's common convention that we use relative change ratios where we normalize the increase in cerebral blood flow to baseline flow. When we look into the literature about studies which have looked at the differences in sensitivity between the vertebral arteries and the internal carotid arteries to a acute bout of hypoxia using Doppler ultrasound, we see that only 4 studies show that the vertebral arteries have this increased sensitivity, whereas 6 have no difference. And there's this proposed mechanism that it's the vertebral arteries have this greater sensitivity to maintain blood flow to the homeostatic regions of the brain, but there's clearly a disparity. And this was mentioned in Dr. Fernandes's talk earlier. Now, we believe there could be something to do with data acquisition and expression that could be contributing to this disparity. Now, why do I say that? Is extracranial arteries are usually measured unilaterally, so only on one side, and then are doubled, assuming that the opposite artery is of the same size or is carrying the same amount of flow. Now, doubling a unilateral measurement we know is going to cause some form of measurement error from the true bilateral measurement, but this hasn't been quantified and hasn't been shown or quantified to show what effect this may have on the interpretation of our data. It's worth noting, and again was mentioned a little bit earlier, that there are differences between the left and right vessels. Particularly, there's reports that the right vertebral artery has 20 to 30% less blood flow than the left artery, whereas the internal carotid arteries seem to have equal flow. This is usually used as the rationale as to why the right vertebral artery is overwhelmingly chosen to be scanned, under the presumption that it will underestimate absolute values of global and posterior cerebral blood flow. But whether this has any impact on the relative response is unknown, and I'll go into that in a second. Finally, not all individuals are going to be smaller or have less blood flow on their right vertebral artery. So should we actually be identifying the vessel with the lowest or highest blood flow if we're using the rationale that we're going to underestimate these absolute values? Now, you may be thinking, well, how might these differences in anatomy or resting blood flow have an impact on relative change ratios? Aren't we already accommodating for these differences in baseline flow by normalizing them or using these percentage change ratios? Some of you may be aware of some work by Atkinson and Batham that they looked into the percentage change response in flow-mediated dilation in the brachial and femoral arteries, where they found this negative relationship between baseline diameter and the percentage change in diameter of flow-mediated dilation. They found as way that this disproportionately scales across the values of baseline diameter, which basically means that vessels with small diameters overestimate the relative FMD response. So we thought we could apply this here and go that intra-individual anatomical and resting blood flow differences within an individual may add considerable error to doubling a unilateral measurement from the bilateral measurement, which may mean that while we're seeing these differences in percentage change and this greater sensitivity in the posterior region could be the product of the fact that we're taking measurements from the smaller right vertebral artery. So we wanted to examine the bilateral internal carotid and vertebral artery blood flow regulation or response to hypoxia, and then investigate and quantify the measurement error between doubling a unilateral measurement and the bilateral measurement of the extracranial blood flow response to hypoxia. So we scanned 44 participants during 90 minutes of normoxia or hypoxia. And a key thing here is that our cardiorespiratory responses, so blood pressure, CO2, and ventilation, were not different during the period of time I was scanning their arteries. It took a little bit of time to scan all 4 arteries, but the cardiorespiratory responses were the same. We saw globally this 30% increase in flow is what we expect with hypoxia as a stimulant. And we actually saw that all vessels dilated to about 7%. When presented as the absolute change in blood flow, we see the internal carotid arteries in gray has this much larger increase in flow compared to the vertebral arteries in white. But when we normalize this to baseline flow, we actually found that there's no differences between the internal carotid arteries and the vertebral arteries sensitivity to hypoxia. We wanted to see if we could kind of match how Atkinson and Batham saw this negative relationship between baseline diameter and the percentage change in diameter. We used flow instead. So we're on the x-axis, we've got baseline normoxic flow, and then on the y, we've got the percentage change in blood flow. And we see that for both the internal carotid and the vertebral artery, we see that arteries with the lower blood flow are associated with a larger percentage increase in the blood flow response. if I draw your attention to the shape of the scatter plots as well, we see that smaller arteries, or arteries, sorry, with the lower blood flow, have this greater variability in response. And this might be seen a little bit more clearly in how we quantify our measurement error. Now, these are Bland-Altman plots, and they're quite busy, so I'll bring your attention just to one to begin with, explain what a Bland-Altman plot is. Bland-Altman plot is a technique, or an analysis that sees how two measurements methods are close to each other. So in this case, we've got doubling a unilateral measurement against the actual bilateral response. And what you've got is you've got the bias, which is the difference between, and we've got the relative response here. So the difference between the two relative responses, and the 95 confidence intervals, which I've then written in red at the bottom of the graph. And then we've got the limits of agreement, which is 1.96 standard deviations, so the spread of the data. For this, then we picked our unilateral measurement based on the lowest or highest blood flow on the left or right, respectively, for the internal carotids and the vertebral arteries at top and bottom. And to try, found that the vessels with the lowest blood flow cause a group mean overestimation to the relative blood flow response to hypoxia, and vice versa. Highest blood flow of that individual cause a group mean negative, so an underestimation. When you look at the range, we see that vessels with the lowest blood flow have a much wider range of measurement error, which means that you should be looking at the vessels with the highest blood flow to limit measurement error. So they've got a thinner measurement error range, and they cause less or underestimation. When we organize this by the more conventional choice of left or right, so we've got the left arteries on the left-hand side, A and C, and right on B and D, bring your attention to the commonly picked right vertebral artery that on a group mean level cause overestimations to the relative blood flow response and to the greatest magnitude. So to quickly give our take-home messages, the first one would be that there's no differences in the sensitivity or regulation between the extracranial arteries, and that they all do dilate to hypoxia. On a group mean level, our unilateral measurement error was quantified at a modest 4 to 5%. And this was particularly in the right vertebral arteries, or arteries which have the smallest blood flow. And this may contribute in some sense why we see the greater percentage increase in the posterior region. And then also, because of the large range of possibilities of measurement error, small sample sizes where these conclusions are being made off of samples of 6 to 8 increase the likelihood of magnitude of error that could be up to 30%, nearly double the expected response. So what do we recommend? Well, change your thought process. Don't select left and right arteries. Select the arteries based on either lowest or highest baseline flow, and we would suggest the highest baseline flow. Because one, it limits measurement error on a group mean basis. They have not as wide limits of agreement. I'd say any sonographer would say that the largest artery is going to be easier to isolate and keep a image of. And then instead of having to go through the process of calculating flow using all these automated softwares, you could use the inbuilt caliper methods to identify the largest flow artery by diameter because of the strong index between flow and diameter. So I'd like to thank everyone who contributed to the study, in particular Dr. Sam Oliver, who is my primary supervisor. So I'd like to take any questions because we would really love some feedback on this.
Great talk, Alex. We have a couple questions here. Before I go to the field, I've got one from the panel. Dr. Hoiland wants to have a go right now.
Hey, excuse me, great talk, Alex. I think first, this is super important and definitely something we all need to be aware of. My one question is, what do you think is the feasibility of in studies where we need to either dynamically measure blood flow and we don't have time to take 4 separate measures? Say you have an individual at an oxygen saturation of 70%, which anyone that's done a study like that will know is quite uncomfortable. What do you think is the feasibility of simply pre-screening people and excluding individuals that have large bilateral differences, such as, you know, anything more than a 10% difference in diameter or something like that, for studies where it might not be feasible to measure everything at once?
Yes, like it's very common now that you see it in the journals that people do pre-screen. So absolutely, you could either base it off of our scatter plots where you see right down, let's say vertebral artery, very, very low flow, less than 50 or 60. You can see you get this greater variability in response. It might be you just pre-screen people that have got very, very low differences, low flow arteries, or like you say, from our data set, I'm pretty sure off the top of my head, the left and right differences, sorry, so large differences were about 100% for the internal carotids, but the vertebral arteries, there was one individual who had 400% difference in flow in one artery. And these are the individuals that you need to be identifying, and they're the ones that are causing the large skews in data. And if, you know, some are going, we didn't pre-screen people beforehand, you're really chancing it because the error that you could be making is very large. So I would say it's why we suggest pre-screening seems to be the in thing, and it's great. Use caliper methods to just get an idea of the size of the vessel and use that as an index of flow and pick the highest one. Of course, bilateral measurements take forever, so we're not saying do that all the time, but definitely try to minimize your error as possible.
Awesome, thank you.
We got one more question from Alex Razumovsky. So what is your opinion on differences of vasomotor reactivity between the VA and ICA? I'm assuming it's, and whether or not you think these high and low flows will impact the error in those measurements.
It's such an interesting one because we've got 44 people in our sample, and when you look at the group mean error, it's only 5%. So you go, okay, well, you know, let's say the differences is only 5%. Is that really going to cause these significant differences? Is it more to it? Whether or not we should actually, I don't know, using, I don't know, the percentage change ratios as the ability to determine these differences. I guess you want to be using the absolute values of O2 usage and delivery, but I appreciate that's so much more difficult. I don't think it's going to have much an effect, but it will contribute. It could be there, but I feel like too many studies are falling pitfall that they might be causing a large measurement error by only measuring one side. So the jury's still out for me. And I feel like if you can do what you can to minimize that error, then, and we start seeing these differences, then yes, we can be more confident.
Great. Another question from Julian Brugnot. Do you think your conclusions would extend to other vascular beds?
Oh, I guess, I think so. I think yourself, Kurt, mentioned earlier, because to do with the selfish brain hypothesis, where there's a lot of individuals who have different morphology in their vasculature. And I feel like, again, if you're making an assumption that everything is equal on both sides, then yes, I feel this would expand further down. If you're only measuring the MCA on one side and you don't know what their vascular inside is going to be like, the other vessel might be doing a compensatory or I don't know if there's an undercompensatory response, but you don't know what the blood flow is on the other side and how this one is going to react. So yeah, I think it would be expanding to the other bed.
Okay, another question from Dr. Anthony Bain. He agrees with screening, or the importance of screening for bilateral differences, but there might be an issue or a risk for selection bias, and we might miss some interesting physiology if we screen them out. Yeah, that's correct. So some of you may be aware of this vertebral artery hyperplasia group that seem to have some interesting morphology, and that was one of our original intents. We're trying to get a big sample of these people. So of course, if you are too stringent with your selection, so if you start saying you can't measure anyone less than 60 mils per min of flow, then you're really biasing it. That's why I think we just want to limit our recommendations to saying within that individual, and to minimize error within that individual, pick the one with the highest flow. Yeah, I think that it's important that this screening process. I was amazed when we did some twins research. We were looking at the left and right of identical twins and seeing that some of the vertebral hypoplasia was actually mirrored. So if it was on the right, it was on the left. And then we did MRI measures and looked at their circle of Willis's, and the lack of symmetry in these twins was quite interesting.
Yeah, on the artery hyperplasia, I think there was a paper by Sato who took them specifically and did them to a CO2 challenge, and they actually found that they have a much what's it called? Not greater, so a reduced response, I'm pretty sure, to CO2. So these are definitely people that you should be looking out for. But okay.
Are there any more questions? If not, I'm going to turn this back over to Dr. Al-Khazraji. She's going to promote our next summer seminar.
All right. Thanks, Kurt. Actually, I'll do the promotion here. Thanks. So coming up on August 5th, we have the exercise and cerebral blood flow, optimizing the stimulus for brain vascular adaptation seminar with a keynote by Dr. Sam Lucas and abstract talks by Rodri Furlong, Gabriella Imi, Sam Weaver, and Lena Shoemaker. And this is at 2 p.m. U.S. Central Time. So we hope to see all of you there.
Yeah, and one more thank you for all of our speakers today. Great questions, great talks.